Hello students, welcome back to the biology class. So in the last class we continued the chapter number 11, the biotechnology principles and processes, and we discussed about the various vectors which are involved in the cloning of genes in plants as well as animals. We learned in the last class that there is a natural genetic engineer which inhabits the soil, and it's a bacterium, and we learned that its name is Agrobacterium tumefaciens, and this is a pathogen which attacks the plants, generally the broadleaf plants which are called as the dicot plants, and induces the formation of crown gall tumors. And this tumor inducing plasmid carries a piece of DNA which is responsible for this transformation of normal plant cells into the tumorous cells, and that piece of DNA is called as the tDNA. We learned that if this disease causing ability of this pathogen can be removed, then it can become a easy tool for transferring the gene of interest, because it will continue to have the mechanism of intervening or entering into the plant cell, and followed by transferring the gene of interest but without causing any infection or without causing any transformation of normal cells, plant cells, into tumorous cells. So that's why we understood the concept of disarmed pathogen vector in the last class. We also learned that in the same manner there are some retroviruses in animals which are able to transform the normal cells into the cancerous cells. So in the same manner, a retrovirus which carries the RNA, so that means we have to talk about here the complementary DNA formed by reverse transcription. So if this virus somehow is made to undergo a change, and it becomes a non-pathogenic type or disarmed pathogen, then it can be used to transfer the gene of interest into the animal cell. So this is also an example of disarmed pathogen vector. So we learned that these modifications are possible in both the tumor-inducing plasmid of Agrobacterium tumefaciens. It is even possible in the retrovirus to carry the desirable genes into the animal cell, and once these gene of interests are joined to this suitable vector, it can be transferred into the bacterium or into the plant cell or into the host animal cell where it undergoes multiplication. Now we learned also in the last class regarding how do we make a host competent to pick up DNA, because it is not easy to transfer DNA. See, what you have learned so far in this paragraph in the last class was regarding the indirect method of transfer of the gene which happens with the help of vectors. But yes, there are methods in which even direct transfer is possible into the cell. It could be bacterial cell, plant cell, or animal cell. So in the last class we learned about that DNA is hydrophilic in nature, so it cannot pass through the middle portion of plasma membrane, because the middle part is the bilayer of lipid, and lipid is hydrophobic in nature. So it is not possible, we cannot easily force bacteria to pick up the plasmid, so we have to make it competent or more eligible to pick up the DNA. So that can be done with the help of treatment with the specific concentration of the divalent cations like calcium. This increases the holes number, and once the hole sizes increases and numbers of holes are created, it becomes easier for DNA to enter inside. And we learned about the trick in this concept, that first we have to expose it to the cold condition, then again a brief exposure to 42 degrees centigrade temperature, again followed by putting back on the ice. This enables the bacteria to pick up the rDNA, and in the case of the animal cells, micro injections are possible, and in the same manner in the case of the plants, biolistics or gene guns are possible, in which gold or tungsten particles, microparticles coated with DNA are made or forced to enter inside the plant nucleus or the animal nucleus, and thus it increases the chances of accepting the piece of DNA. Otherwise, the best method is to use disarmed pathogen vector, which allow the cells in fact to pick up the recombinant DNA because of the parasite or pathogen carrying it into the host cell. So we covered the topic of various tools and techniques required. Now today we are going to start with the process, how exactly we go about, because we have learned about the technique, the theory portion, and we have learned about the requirements which are needed for creating a rDNA. So now today we are going to understand how exactly we are going to create an rDNA. Remember, this will be more like a summary of the whole concept, except for the PCR concept.